Good morning, students. I am Republica Borgohain, Assistant Professor, Assam Downtown University, Department of Pharmaceutical Science. So today I will be discussing about the deficiency of essential fatty acids. So the deficiency of essential fatty acids results in phrenoderma or toad skin that is characterized by the presence of horny eruptions on the posterior and lateral parts of limbs, on the back, loss of hair and poor wound healing. The non-essential fatty acids, they can be synthesized by mammals, so it is not essential to take them in diet. They include saturated fatty acids and monounsaturated fatty acids. Now, then phytanic acid 3,7,11,15 tetramethyl palmitic acid. It is present in milk lipids and animal fat. Then cyclic fatty acids, that is the lipoic acid. It is a hydrogen carrier and coenzyme in oxidative decarboxylation of alpha ketoacids, for example, pyruvate and alpha ketoglutarate dehydrogenase complexes. Now, discussing about the triacylglycerol, that is, all the commercially important fats and oils of animal and plant origin consist exclusively of the simple lipid class triacylglycerols. Esters of fatty acid with the trihydric alcohol glycerol. Glycerol with one molecule of fatty acid is called monoacylglycerol. 2 molecule of fatty acid is diacylglycerol. 3 fatty acid is triglyceride. Fats as stored fuel. Triacylglycerols are the most abundant group of lipids that primarily function as fuel reserves of animals. Fats primarily occur in adipose tissue. Adipocytes of adipose tissue, they predominantly are found in the subcutaneous layer and in the abdominal cavity and are specialized for storage of triacylglycerols. Simple triacylglycerol contain the same type of fatty acid residue at all the 3 carbons. Mixed triacylglycerols are more common. In general, fatty acid attached to carbon 1 is saturated, that attached to carbon 2 is unsaturated, while that on carbon 3 can be either. That means it may be saturated or it may be unsaturated. Now, the properties of triacylglycerols. Hydrolysis. Triacylglycerols undergo enzymatic hydrolysis to finally liberate free fatty acid and glycerol. The process of hydrolysis catalyzed by lipases is important for digestion of fat in GIT and fat mobilization from the adipose tissue. Then secondly, the property is saponification. The hydrolysis of triacylglycerol by alkali to produce glycerol and soaps is known as saponification. That is, triacylglycerol plus 3 sodium hydroxide gives glycerol plus 3 RCOONA, that is soaps. Now, the term rancidity. It is the term used to represent the deterioration of fats and oils resulting in the unpleasant taste. Fats containing unsaturated fatty acids are more susceptible to rancidity. Rancidity occurs when fats and oils are exposed to air, moisture, light, bacteria, etc. Hydrolytic rancidity occurs due to partial hydrolysis of triacylglycerol by bacterial enzymes. Oxidative rancidity is due to oxidation of unsaturated fatty acids. This results in the formation of unpleasant products such as dicarboxylic acids, aldehydes, ketones, etc. Rancid fats and oils are unsuitable for human consumption. Now, what are antioxidants? The substances which can prevent the occurrence of oxidative rancidity are known as antioxidants. Trace amounts of antioxidants such as tocopherols, that is vitamin E, hydroquinone, gallic acid, and alpha-naphthol are added to commercial preparations of fats and oils to prevent rancidity. Now, what is lipid peroxidation? In the living cells, lipids undergo oxidation to produce peroxides and free radicals which can damage the tissue. The free radicals are believed to cause inflammatory diseases, cancer, atherosclerosis, etc. It is fortunate that the cells possess antioxidants such as vitamin E, urate, and superoxide dismutase, that is SOD, to prevent in vivo lipid peroxidation. So now discussing about phospholipids. So they are polar or ionic compounds that are composed of an alcohol that is attached by a phosphodiester bridge to either diacylglycerol or sphingosine. Now the classification of phospholipids. Phospholipids are classified into two classes. That is glycerophospholipids. They have glycerol as a backbone, and spignophospholipids, that is spignomyelins, they have sphingosine as a backbone. So this is the structure of phospholipids. Now the properties of phospholipids. They are amphipathic molecules, that is each molecule has a hydrophilic, that is polar head formed of the phosphate group and the alcohol group, and a hydrophobic, that is non-polar tail formed of glycerol or sphingosine and the hydrocarbon chain of the fatty acids. So now there are different types of phospholipids, but the discussion of the phospholipids, now we are going to say that is the functions of phospholipids. So phospholipids, they participate in the lipoprotein complexes, which are thought to constitute the matrix of cell walls and membranes, the myelin sheath. The role in enzyme action is certain enzymes, they require tightly bound phospholipids for their action. For example, mitochondrial enzyme system involved in oxidative phosphorylation. Then role in blood coagulation. Phospholipids play an essential part in the blood coagulation process required at conversion of prothrombin to thrombin by active factor X. Possibly also in the activation of factor 8 by the activated factor 9. Now its role in lipid absorption in intestine. Lecithin lowers the surface tension of water and aids in emulsification of lipids, a prerequisite in digestion and absorption of lipids from gastrointestinal tract. Now its role in transport of lipids from intestine. Exogenous triglyceride is carried as lipoprotein complex. Then chylomicrons, in which PL takes an active part. So there are the several rules we can say, and there in my next video I will be going to discuss about the other parts of the lipids that act as the basic sources of lipids. So here lastly I want to conclude about what is lipoproteins. So lipoproteins, there are different types of lipoproteins are there. That is chylomicron, that it transports dietary triglyceride and cholesterol esters from intestine to peripheral tissues and liver. Then very low density lipoprotein, that is VLDL, that transports endogenous triglyceride from liver to extrahepatic tissues. The low density lipoprotein, that is LDL, it transports cholesterol from liver to extrahepatic tissues. Then high density lipoprotein, that is HDL, it transports cholesterol from extrahepatic tissues to the liver in an esterified form. Since lipids are water insoluble, they are present in the blood in the form of lipoproteins, which are water soluble. They are composed of triglyceride and cholesterol ester core surrounded by a cell of proteins, also called as apoproteins, phospholipid and free cholesterol. They are separated by ultracentrifugation into 4 distinct groups based on their density and by the process electrophoresis. So this was a simple discussion regarding the lipid. So here I end up with the discussion regarding lipids, and in my next video I will be discussing about what are carbohydrates and their functions. Thank you.